Prashant sir, you are. Good morning, everyone. A warm welcome to everyone in today's session. This is Sanskriti Tiwari Chaudhary, Assistant Professor, Microbiology Department, Parul University. Today's webinar topic is on Western diet, microbiome, and bile acid linked to systematic inflammation and early signs of Alzheimer disease, which will going to be delivered by Dr. Prashant Kumar Jaina, sir. Right? So, before going to start the session, let me first give you the brief introduction of Dr. Prashant Kumar Jaina, sir. Sir has done his postdoctorate from the University of California. University of California awarded him Outstanding Researchers Award. He has co-authored and authored several international papers as well. He also wrote many book chapters which was published in Nature, a big achievement for a scientist, right? So after then, he is all having more than 10 years of research experience in studying the impact of diet and gut microbiota on the development of metabolic disease, which includes obesity, diabetes, and carcinogenesis. Presently, he is working as the capacity of project scientist in Department of Pediatrics, Cedar Sinai Medical Center, LA, Los Angeles. Right? So, without wasting much time, because the audience are highly eager to hear from you, sir. So, now moving to Dr. Jaina, sir. So, hello, sir. Audience are highly eager to hear from you.
Okay, good morning, everyone. So, thank you very much for giving me an opportunity to present my work. So, I am delighted to interact with various students and faculty members after this presentation. So, hopefully, you guys like my work. Okay, so currently I am working on different cardiovascular disease model, but I will show you my work on Western diet, how the Western diet change your lifestyle and your microbiome profile, and it causes different kind of inflammations and slowly lead to your Alzheimer disease. So this building is of Cedar Sinai Medical Center at Los Angeles. So where I work. So let's start my work. So my work will focus on Alzheimer disease. What kind of Alzheimer disease is this, and how it work, and what kind of inflammations you observe. Different kind of diets can cause Alzheimer disease or inflammations. There is a Western diet that changed your microbiome profile, bile acid profile, and cause inflammations. Then you can change your microbiome profile or remodel your microbiome to improve your gut health. That can reduce the Alzheimer disease risk. So here we can start with the people living with dementia around the world. So you can see the dementia is a kind of Alzheimer disease. In 2020, Alzheimer disease is the 6th leading cause of death in the United States. More than 5 million Americans are living with Alzheimer disease. You can say 16 million Americans provide unpaid care of people with Alzheimer for the dementias. So if you see here, if you see here, the people living with dementia is Asia is 67.2 million in 2050. So, and in around the world, you can see around 131 million people will suffer from Alzheimer disease. So, and very few drugs are available for the treatment of Alzheimer disease in the long run. So this is a challenging task for the people and for the researcher to know about the Alzheimer disease and how we can cure for this. So Alzheimer disease symptoms is the loss of memory, cause confusion, having poor judgment, vision problems, and social changes, and also it loss of memory. So is older disease people suffer after old age, after 50 or something, but nowadays is very challenging. Those who are suffering from cardiovascular disease or diabetes, obesity, they also can leads to Alzheimer's disease. So as I'm presenting mostly audience from researcher from India, so let's think about more Alzheimer's disease prevalence in India. After China and US, India is the third leading cause of Alzheimer's disease around the globe. More than 4 million people have suffered from dementia in India. And most important is Western diet is linked to Alzheimer's disease, and especially the consumption of meat, sweets, and high-fat diets, products that characterize the symptom of Alzheimer's disease. So changing habits, changing eating habits of rapidly changing the nation. So you can see here a pie chart saying that in 1961, many of us eat grain, few sugar, dairy products, egg, but in 2011, the produce increase, dairy and eggs increase, sugar increase more. So slowly the dietary patterns changing. So the behavioral risk also changing with the dietary factors. So high blood pressure, environmental risk, cardiovascular risk, they all changes due to this dietary burden. So recently, there is a paper, it came in Lancet, they said that high intake of sodium and low intake of whole grain and low intake of fruits, the leading risk of death and dementia. So it means that if your daily dietary habit changing your lifestyle and it causing a slow disease that can lead to your major disease like Alzheimer's disease. So let's look into what kind of diets are available commercially for the research. So mostly people use chow diet, which is a regular diet, control diet comparably with chow, and we have a Western diet and high fat diet. So this Western diet, you can see more amount of carbohydrate, fat, and less amount of protein. So more fat and sugar that causes the diet that we are getting. So what are those? So these are delicious Western diet. We all know what these diets are. They are very delicious and same time they can cause lot of chronic diseases. So I show you some in red highlights. Those are in my work. I will show you later on. But it causes obesity, liver disease, Alzheimer, and psoriasis, kind of deadly diseases as well. So Western diet generally classified with high calorie, high fat, sugar, can cause Alzheimer's disease and other chronic diseases. Western diet induce systemic inflammations, microglial activation and reduced neuroplasticity. That is one of my work. Mediterranean in diet, like more low intake of meat and high intake of fruits and fish and vegetables, they reduce the risk of Alzheimer's disease. So, so from this particular information, we can work on that, how we can manipulate the dietary habits and change our lifestyle. So starts my work, see the Western diet induces gut dysbiosis. So dysbiosis means change your microbiome profile inside your intestine. So you can see the orange one is control diet, and the Western diet is the green one. So they have a different microbiome profile. So when you see the Firmicutes to bacteroids ratio, they are high formic to bacterial ratio in Western diet. So mostly the obesity and inflammatory people or mice model, they have an increased amount of formic to bacterial ratio. Proteobacteria, mostly the gram-negative bacteria is the inflammatory marker, also increase, and actinobacteria is mostly beneficial bacteria is decreased. So at the genus level, we see that Bifidobacterium, including with other bacteria, is reduced. So the Bifidobacterium is one of the good bacteria that resides in the intestinal tract, and Bifidobacterium helpful for yearly development of kid. So, and there is some gram-negative bacteria like D-sulfovibrio, they are increased. So from this slide, we can say that, so when you eat Western diet, there is change in microbiome profile. So Western diet induces systemic inflammations. Systemic inflammations means you can get the inflammation or inflammatory marker around the body. So here I'll show you some inflammatory markers in the brain. In the microglia, it means there is a cell in the hippocampus region, and we isolate the microglia. And from microglia, we saw that the similar inflammatory marker like IL-1 beta, IL-6, TNF-alpha, those markers are highly induced in the brain microglia as well as in the spleen. So spleen, you can see the total macrophage, TNF-alpha macrophage, they are increased. They are mostly inflammatory markers. And you can see here, CD11B is a macrophage marker. Is also, we can see the green dots in the fluorescence imaging, more green dots here in the Western diet. So here, those inflammatory markers are highly present in the brain and microglia cell as well as in the spleen. So it's causing Western diet, changing microbiome, changing inflammations, and also Western diet feeding reduces long-term potentiation, means how your memory intact. So when you eat long-term Western diet, Western style diet or Western diet, these mice models has a reduced memory. Long-term potentiation means when we isolate hippocampal cells and we give electrophysiological techniques, measure this potentiation. Those who have a more memory, they have a higher signaling, and those who have less memory, there's a lower signaling. So here, if we eat those kind of diets with changing memory profile, it reduces long-term potentiation. At the same time, we see those postsynaptic density 95 is a marker of memory, and phospho-ORC and total phospho-ORC is the marker of inflammations. So inflammations are increased in Western diet, and PSD95 is decreased in the Western diet. Similarly, the brain-derived neurotrophic factor, they reduce in the brain, microglia, liver, and adipose tissue. So from here, we see that Alzheimer's disease is a symptoms where we reduced memory, or it's memory decline, or it declined the thinking, or declined the reasoning skills. Similarly, in Western diet-fed mice models, also reduced long-term potentiation and as well as the memory. So one of in my headlines, I showed that in Western diet change microbiome, change inflammations, and also it changed bile acid profile. So this Western diet mice model, it's easy to build, is a specific pathogen-free wild-type mice, C57 mice, and we supplemented Western diet having high fat and high sugar for 9 months. At the 10th month, we sacrifice them, and we see those mice liver are completely whitish, and there's a lot of fat droplets in the liver. Then we see the brain, liver, and serum bile acid profile, and we saw certain bile acid like deoxycholic acid, taurocholic acid, and certain other beneficial bile acid are changing. So these bile acid are stored in the gallbladder, synthesized in the liver, and circulate to the intestine and come to the liver again. So per day, approximately 37 times it circulates. With the help of 37 different enzymes, it circulates from the intestine to enterohepatic circulation to the liver. So it keep on circulating. It helps in metabolism and all this. So when there is excess amount of bile acid, that leads to cancer or other metabolic disease. So here in this Western diet model, we see deoxycholic acid and taurocholic acid is increased, and other chenodeoxycholic acid, orthodeoxycholic acid, and taurolithocholic acid, those are reduced. So what happened to the metabolisms? So here we saw the certain bile acid receptor gene like farnesodiol receptor, SSP, PFR gamma in the liver, ileum, adipose tissue, and brain. We all see that FXR, those is selectively a bile acid target gene, got reduced in Western diet. It means there is a maximum amount of bile acid deposited in the liver. So bile acid changing. So what happened? So we use a chemical. It's called cholestyramine. So what happened? We at the 7-month age, we supplemented cholestyramine for 3 months. So cholestyramine will clean your bile acid deposition in the gallbladder. So it's wipe out. So when it wiped cholestyramine, we saw those inflammations are induced in brain, those got reduced after cholestyramine. It means bile acid playing a role in causing inflammations. When we give cholestyramine, the inflammation got reduced in the brain. You see the brain-derived neurotrophic factor that reduced in the Western diet model got selectively increased. Certainly CCL17 and some potassium channel, IL-1 beta, they got reduced in cholestyramine supplementations. So bile acid playing a role. So here we focus what this bile acid is for. So bile acid is a chemical synthesized in the liver and stored in the gallbladder and playing a role in obesity, circadian rhythm, glucose metabolism, insulin sensitivity, and all this. So here, see, there is a certain bile acid receptor called TGR5 or G protein coupled bile receptor 1, the TGR5, the same name. The TGR5 is present in the gut, the intestine, as well as in the brain. And it plays at that gut microbiome and brain relationship. So this will in the future will target on this particular TGR5 signaling and see how it works in the Western diet model. So this bile acid homeostasis is regulated by liver and intestinal enzymes. So this slide is very crowded, but this published in my previous lab says that it has a liver have a certain enzymes and intestinal lumen has a certain enzymes. So this deoxycholic acid and lithocholic acid, these are secondary bile acid. So usually the primary bile acid is synthesized in the liver, and the secondary bile acid is converted with the help of gut intestinal bacteria. So, and this bile acid is play a role in liver cancer, GI cancer, obesity, immune response, and other infectious disease as well. So a report came in Cell Press is saying that the metabolic network analysis reveals altered bile acid synthesis and metabolism in Alzheimer's disease. So you can see the bile acid is synthesized from the cholesterol, and here the several bile acid like deoxycholic acid, lithocholic acid, they play in the brain region and causes transcriptional regulatory. So, and the another group in Duke's Institute of Brain Science, they reported that significant elevation of certain bile acids such as glycodeoxycholic acid, taurodeoxycholic acid, taurolithocolic acid, and glycolithocolic acid is found in human brain samples. Researchers also found that the deoxycholic acid and lithocolic acid, deoxycholic acid is found in the brain, which help in 7-alpha dehydroxylation of cholic acid by the gut bacteria. So here, the human sample and the mice model, they both have increased deoxycholic acid, and those formation happens due to the gut bacteria. So what gut bacteria play? Gut microbiome play several roles. Like, in the stomach we have a pH 1.3. Still we have a several amount of bacteria like lactobacillus, streptococcus. In a duodenum, increase a little bit more bacteria. Here we can find lactobacillus, streptococcus. Jejunum we have a bacteria. Colon we have a more amount of bacteria like bacteroids, Eubacterium, Clostridium, Bifidobacterium, all these. So different pH, those bacteria reside in your intestine, and they play a role in central nervous system, short chain fatty acid, fat storage, protect against pathogen, synthesize vitamins, it change in immune response. Lot of beneficial role it play in the intestine and in our health. So as I am focusing on microbiome and Alzheimer's disease, which is a old age disease, you can see that young to old, here more amount of bacteroids when they are young, and slowly formicutes are increasing and the bacteroids are decreasing, and some other bacteria is popping up, like proteobacteria is popping up here, more amount of proteobacteria when you are older. So is less amount of proteobacteria, slowly it increases and keep on increasing. So proteobacteria is a gram-negative bacteria which causing inflammations. The intestinal bacteria play a crucial role in host aging process. Health span promoting interventions are accompanied by normalization of gut microbiota. So microbiota targeted intervention may be a promising target for anti-aging or Alzheimer's disease. So microbiome is most important part of your health. So particularly when in India, then high antibiotic uses. So literally, when you go to a doctor or a physician, either you say you are cough or you say you have a fever, you will get antibiotic for sure. So antibiotic are a group of drug. It causes it kill bacteria. So and sometimes the patient is recovered from either from infection or fever or whatever symptoms he has because of antibiotics. So it means there is a lot of opportunity for looking at why consumption of antibiotic will heal the disease. And also same time we have to reduce the uses of antibiotics. So that's why study on microbiome is most important to look around various diseases. So what microbiome play a role in Alzheimer? So various research came such as probiotic supplementation reduce Alzheimer disease. Alzheimer disease microbiome is associated with the dysregulation of anti-inflammatory P glycoprotein pathway. Fecal microbiota transplantation reduce Alzheimer disease in mice model. So several people were working on how microbiome remodeling will help reducing Alzheimer's disease. So recently one paper came, one study came from China. They published that oligomanate, a Chinese approved drug, FDA approved drug for Alzheimer's disease after 17 years. What is the drug? The sodium oligomanate therapeutically remodel gut microbiota and suppress the gut bacterial amino acids shaped neuroinflammation to inhibit Alzheimer's disease progression. So this sodium oligomanate will help in reducing Alzheimer's disease by changing microbiome profile. So after this study, we are very much interested to look around what kind of microbiome we can change. So what kind of microbiota play a role in Alzheimer's disease and causes inflammation and reduces memory. So we will implement all those study in our research. So in one of my study, I did, okay, let's see, if microbiome is playing a role, let's remove microbiota by using antibiotic and see what it affects. So similarly, as like cholestyramine, I provided 3 months. So here I give Western diet for 10 months. And when I completed 7 months, I started antibiotic like ABX is ampicillin, vancomycin, metronidazole, and neomycin. Vancomycin alone and polymyxin B, these are different target. Like ABX is a broad spectrum antibiotic. Vancomycin is only target gram-positive bacteria. And polymyxin B is target gram-negative bacteria only in the drinking water for 3 months through the mice model. So what we see that after supplementing drinking water antibiotic, there is change in microbiome profile. They diverse. You can see the change in microbiota. The diverse microbiome has splitted. With antibiotic, they completely separated. With the vancomycin, they stick in one place within all other groups. But in polymyxin, in the vancomycin, they completely separated from the groups. So if you give broad spectrum antibiotics or you give gram-positive bacteria, they are completely separate. So in the diversity, the beta diversity also, it says that they have a completely different microbiome profile between controlled diet and Western diet. When after supplementing antibiotic, they reduce the diversity. So some phylum level bacteria also changed. Here we see supplementing with polymyxin B, they kill all gram-negative bacteria, but the actinobacteria, which is beneficial one, having bifidobacteria, within the actinobacteria, they increase much here. Proteobacteria, when you give only gram-positive bacteria, target vancomycin, all gram-negative bacteria got gram-positive bacteria got killed, but gram-negative bacteria survived. So certain like D-sulfovibrio were reduced. So within this antibiotic, we saw changing in microbiome profile as per the hypothesis. So then we see certain inflammatory genes like IL-1 beta, IL-6, TNF-alpha, ApoE, and some calcium channel blocker. Those are significant marker of inflammation in the Alzheimer's disease. They have got reduced in antibiotic treatment. So here we are thinking that the antibiotic, when you kill the antibiotic, it helping reducing Alzheimer's disease risk by changing microbiota profile. So same as in Western blot, we see that IL-1 beta got reduced in ABX treatment and polymyxin B, but not in vancomycin. So mostly here we see that the broad spectrum antibiotic and targeting gram-negative bacteria is changing the inflammations, but not in vancomycin treatment. So in IL-1 beta, we didn't see much change in the brain cytokines profile, but in TNF-alpha, we saw it reduces in vancomycin and polymyxin B. It means it varies in different results that antibiotic is reducing your brain inflammations. So then we thought, okay, if gram-negative targeting bacteria or broad spectrum bacteria is healing inflammations, then we supplement bifidobacterium. So because the bifidobacterium is reduced in Western diet, and when you supplement, you target gram-negative bacteria like polymyxin B, is mostly the actinobacteria is increased. So we thought bifidobacterium might be a target to see how it works. So previously I published that the bifidobacterium infantis are helping in non-alcoholic steatohepatitis in Western diet model. So I thought we'll target to the Western diet mice model to see how we can reduce the brain inflammations. So the probiotic bifidobacterium infantis lower intestinal inflammations, is a healthy probiotic strain. It normalizes intestinal microflora, stimulate intestinal motility, support colonic health, improve immune systems. There's a lot of beneficial effects. So the gut dysbiosis, restoration by probiotics. So we supplement probiotics. How we supplement? Same strategy. We Western diet for 10 months. So we supplement bifidobacterium infantis, 10 to the power 8 CFU per dose oral GABAs. So we see a change in microbiome profile. We have increased actinobacteria. We saw here. We saw there is an increase in Bifidobacteriaceae, the Bifidobacterium strain here after supplementing Bifidobacteria. The Western diet model, it increased formicutes to bacteroids ratio, but after supplementing B. infantis, it got reduced, and the Bifidobacterium supplementation increased the profile here. And the gram-negative target, gram-negative inflammatory D-sulfovibrio got reduced. So we saw that, okay, in Western diet model, Bifidobacterium infantis changing microbiome profile and increasing the Bifidobacterium strain that reduces inflammation and improves memory. So I show you some inflammatory genes and proteins. IL-1 beta, IL-6, and some potassium channel, ApoE, those are increased in Western diet, got reduced in Bifidobacterium infantis. Similarly, the IL-6 got reduced with B. infantis, phospho-ORC got reduced, and CD11B, the macrophage marker, also reduced in B. infantis treatment. So both in gene and in Western blot as well as in brain tissue homogeneous cytokine profile, we saw that reduced IL-1 beta and TNF alpha. So it consistently reducing inflammation with B. infantis. So how about memory? We saw that the control diet has increased long-term potentiation. When we supplement Western diet, it got reduced, and after supplementing B. infantis, the reduced long-term potentiation got restored at age like normal. So it improving. So here we saw the figure that it says that Western diet, when we supplement with B. infantis, is kind of improving the memory as well as the brain-derived neurotrophic factor and PSD 95, both in RNA level and protein level, they got improved in B. infantis. So this data shows that the B. infantis is working well in Western diet model, and it improving memory and reducing inflammations. So then we look into the bile acid profile. So the bile acid signaling, so I told you that TGR5 or G protein bile acid receptor have improved both in RNA and protein level very significantly, and those bile acid are increased due to Western diet like deoxycholic acid, taurocholic acid in Western diet model. After supplementing probiotic B. infantis, they got reduced. So here it shows that both in Western diet and supplementing B. infantis, the bile acid profile changes, and bile acid receptor like TGR5 and FXR, they got activated and improving memory and reduces inflammations. So when we give more Western diet to the mice, here we saw cholesterol metabolites, in the cecal metabolites, cholesterol got increased, cholesterol level. So from cholesterol, bile acid synthesized. So when we supplement Western diet with bean fatties, the cholesterol that got highly increased metabolites in the cecum, they disappeared here in the Western diet with the bean fatties model. So it says that bean fatties is nicely clear the cholesterol deposition in the brain as well as in the intestine, and reduces the bile acid deposition in the gallbladder. So in another analysis of the cecal microbiota, we saw that the Western diet model has a reduced unsaturated fatty acid and reduce purinones, the amino acids. But after supplementation of bean fatties, we have increased saturated fatty acid. So that will helpful in the colonic absorption of different nutrients and healthy gut. So here I can tell you that the gut dysbiosis and dysregulated bile acid profile synthesize may play a role in early onset of Alzheimer's disease, because we saw a lot of loss of memory, more inflammation of Alzheimer markers, and increased proteobacteria, and decreased bifidobacterium, and the bifidobacterium are good, healthy probiotics, reduced bile acid receptor signaling in Western diet model, and also other mice model and human samples. Reduced harnessed X receptor and TGR5 signaling, like the G protein bile acid receptor, as well as the bile acid ligand, such as chenodeoxycholic acid, orthodeoxycholic acid, and triurolithocolic acid. Those are significant ligand for FXR and TGR5. There is certain toxic secondary bile acids, such as deoxycholic acid and taurocholic acid, that are increased in Western diet. So what are the treatment strategy? We implement the microbiome, which manipulate microbiome or remodel microbiome in Alzheimer's disease pathobiology. Then change intestinal microbiome to restore long-term potentiation and memory. Then bean fatties is one probiotic which restore microbiome dysbiosis. There is a certain other potential probiotics as well, and there is certain commensal bacteria that got reduced due to Western diet. We may target all those probiotic which may helpful. Then bile acid sequestrant, that is a cholestyramine, that is FDA approved drug for liver disease, and also can be implemented to see the reduced inflammatory response in the brain. So this I will collaborate a lot of people for this work. So with this, I worked with my previous lab is Yvonne One, where I implemented all these studies. Then thanks to David Mills, who help in early starting microbiome analysis. Dr. Krish Krishnan, who is helping the large data integrations. Li Wei, Jean, who help in brain studies, electrophysiologies. Izumi and Jacopo, they are all the Alzheimer groups. They helping electrophysiology studies and hippocampal isolation, microglia studies. Then these are my Lily, Inghu, and Huisin. They are my lab member. Nidhi is another student who work in my lab for the studying the liver pathology of this. Currently, I am working with Mose RDT for studying atherosclerosis and Kawasaki disease with Magali is my group leader. So a lot of people and a lot of funding agencies helping on this study. So within this, I thank to Parul University, Gujarat for helping for giving me this opportunity to present my work. So I am happy to have any questions. Thank you.
Yes, sir. So some audience they texted me, right? So first question they asked like that you talk about Western diet, right? So what will be the
Yes.
composition of Western diet? Am I audible
Okay.
to you,
So
sir?
yeah. So the Western diet, I showed the composition. So I will go back to the slide again. Uh, so the Western diet has a more amount of fat. So here, the protein is a 7.3 by weight. In percentage, carbohydrate, fat, fiber, and in kilocalorie, the protein is 15.5, carbohydrate is 41.8, and fat is 42.7. So this is the Western diet I used.
Okay, sir. Thank you. So the next question is whether it's a microbiota or Western diet or a combination of both are a causative factor for Alzheimer. Can the impact of one factor is reversed by the other?
Likely, so I showed you in both way. One is how diet changes memory, then how microbiome improve memory. So this is a two-way. So if we supplement directly anti the gram-negative bacteria like proteobacteria to the mice model, they have a more inflammatory response. Maybe there's some kind of intestinal inflammations, but it's very interesting to see individual bacteria effect or individual diet effect. So I think they work together because intestine bacteria want nutrients to grow. If we will not provide any diet, they will die right there. Okay. So we have to eat food to grow, right? Or eat food to survive. So then only microbiota will digest our food and supply energy to our body. So that's we go both way.
Okay, sir. And the third question is, sir, as despite we are taking so many control measures while we performing our experiment, but as we already know that every individual mice has a different immunogenic response. So how can you justify the result of your experiments? Because every mice having their own immunologically responses, right? So how
No,
will you
yeah,
going to justify?
I agree with you. So each mice in each different colony, in different housing environment, they have a different immune response.
In
But
different conditions
consistently,
as well, right?
it what?
In different in conditions as well,
Yeah,
environmental
in different
conditions.
environment and different housing, they have a different microbiome profile, and they have a different immune response as well.
Okay.
So at one point of time, we can see that how Western diet to changing your microbiome profile or bile acid profile. So I showed you some study they published in North Carolina, the Duke Institute of Brain Science. They showed that human Alzheimer disease has a deoxycholic acid. And I studied my work in California. That is maybe 6 hour flight drive to North Carolina in one coast to another coast, and I saw that Western diet having similar amount of deoxycholic acid. So it means that it's consistently Western diet causing or changing bile acid profile. That is for sure. In that context, in different environment, there is some changes. That is for sure. But we have to find out if that changes is protecting or if that changes is causing more serious diseases. So maybe mice model is not the perfect model for that condition. But at the same time, there is certain transgenic mice like 5X FAD mice and some other PSEN, APP knockout mice are there to mimic the Alzheimer's disease risk with those mice.
Okay. Am I audible, sir?
Yeah, yeah,
Yeah, okay, sir. Connection was lost actually. Okay, sir. So, am I audible now, right?
yeah, yeah.
Am I audible?
Yes, you can see my screen, right?
Yeah, yeah. Now I'm able to see because, sir, actually connection was lost here. So, because of this reason. But, sir, you are showing that a screen commercial diet for research as per human consumption, right?
Mm-hmm.
Now you are visible and audible to us. Due to connectivity issue, we facing issue. So I hope audience you all enjoyed Prashant Jaina sir session. Obviously, yes, they are saying to me here in inbox. So audience, if you have any question, any queries related to this particular topic, you can drop me your message. You can WhatsApp me or mail me. I will forward your message and question to Prashant Jaina sir, and then I'll revert back to you. Fine. So, sir, we, the Paro
Yes.
University, are highly privileged that you presented between us, and we are grateful for your precious time and obviously for you with your deep enlightenment, right? So, we are highly privileged. So, thank you, sir.
Thank you.
Thank you, sir. Thank you. Sir, you can disconnect.